Hello everyone, we are Group 16. Today, we'd like to talk about alpha-beta T-cell receptor. What is receptor? Immune system is divided into two types, innate immunity and adaptive immunity. Adaptive immunity provides specific protective immunity and it prevents reinfection. Lymphocytes make this possible through the antigen-specific receptors on their surface. Each lymphocyte has receptors of a single specificity, so the population of lymphocytes recognizes a great variety of different antigens. What is the function of T-cell receptor? Lymphocytes are roughly divided into two types, B-cell and T-cell. The antigen-specific receptor of B-cell, called B-cell receptor or BCR, can be secreted as antibody. BCR and antibody recognize antigens in the extracellular spaces. However, the antigen-specific receptor of T-cell, called T-cell receptor or TCR, is never secreted. In this case, antigens are broken down into peptides by proteases, and one of the peptides is delivered to a self molecule called MHC molecule. As a result, TCR recognizes both peptide and MHC molecule and it activates T-cell. What is the structure of T-cell receptor? TCR is a heterodimer consisting of two different chains called alpha chain and beta chain. These two chains are linked by disulfide bond. Each chain is composed of two regions called variable region or V region and constant region or C region. V region of alpha and beta chains together form the antigen binding site. A minority of TCRs is made up of different pair of chains called gamma chain and delta chain, but the function of these receptors is not well known. How is it built? The diversity of V regions is generated by two main processes, combinatorial diversity and junctional diversity. V region is encoded by separate gene segments called V, D, and J gene segments. There are multiple copies of these gene segments, and it is a random selection of just one gene segment of each type that produces a great diversity of V regions. Specifically, alpha chain locus consists of 17 V gene segments and 61 J gene segments, whereas beta chain locus consists of 52 V gene segments, 2 D gene segments, and 13 J gene segments. Each of the different alpha chains could be combined with each of the different beta chains, and it finally makes up around 5.8 million different V regions. This process is called combinatorial diversity. In addition, when different gene segments are joined, random numbers of nucleotides are deleted and inserted at the ends of gene segments, and it generates 200 billion junctional diversity. As a result, the combination of combinatorial diversity and junctional diversity generates a quintillion different TCRs. Here is a question. The answer is on the bottom right corner of the first slide. Thank you for watching.